Good morning, students. Today we are going to learn about in semester 2 introduction to microbiology unit 3. The first topic, general characters of bacteria. The morphology. Bacteria displays diverse morphologies like it has different structures like spherical structure, rod-like structure, spiral. We all name the 10 type of cocci, which is a spherical shaped bacteria, example streptococcus, and bacilli, which is a rod shaped bacteria, example E. coli. Spirella, which is a spiral shaped bacteria like Helicobacter pylori. Like Vibrio, it is a curved rod shaped bacteria seen in Vibrio cholerae, and Spirochetes, which is a corkscrew shaped bacteria including Trypanomalipidium. And the filamentous, filamentous means we all know that it's a long thread like bacteria, example actinobacteria. And the pleomorphic bacteria which undergoes the same species like Coryn bacteria, diphtheria. Next, these are the structures we have learned in the first slide. This is the round shape. This is the in the round shape we have different types, which means single round, and if two cocci are attached, this is called as the diplococcus. Is the more than two, this is called as the streptococcus. These are the tetrads, and the step is the staphylococcus. These are all the coccus, and these are the rod shaped bacteria. These are the spiral shaped bacteria, and these are the filamentous bacteria. These are the pleomorphic, and these are the appendaged bacteria, and these are the box shaped bacteria. The metabolic diversities of bacteria are the phototrophs, which take the light energy through the photosynthesis, and as also in plants we can see as the cyanobacteria. And the chemotrophs, which means they obtain energy from the chemical compounds, and they are subdivided into two types, chemoorganotrophs and chemolithotrophs. In chemoorganotrophs, they utilize organic compounds such as sugars and the amino acids. In chemolithotrophs, they utilize inorganic compounds like sulfur or ammonia. And next, heterotrophs, they depend on external organic compound sources obtained from the other organisms. And next, the autotrophs, they synthesize organic molecules from the inorganic sources, primarily carbon dioxide. They further also divided based on their oxygen requirements, their metabolic end products, which is also called as the fermenters, and by the ecological roles, highlighting their metabolic adaptability and ecological importance across their diverse habitats. And in bacteria, they undergo reproduction in different types, like they reproduce primarily through binary fission, which is a form of sexual reproduction where the single parent cell divides into two identical daughter cells. This process involves several steps. First step, duplication of genetic material, cell elongation, and the septum formation, and formation of daughter cells, and the cell wall synthesis. We can this entire process we can observe in this picture. In this picture we can observe this is the parent cell. Actually this parent cell have single DNA. After some time, when it undergoes the reproduction, the nucleoid which is present in the cell replicates, copy itself. It replicates. This is the structure of the cell. Bacteria, this is the cell wall, this is the cytoplasm, and this is the nucleoid, and this is the replicated. They copied itself. They copied their DNA into two. After that, the cytoplasmic membrane elongates. This is the second step. This is the cytoplasm. This cytoplasmic membrane elongates and separating the DNA molecules. In the first one, we can observe these are some of them are attached, but in the second step they got separated, and the cytoplasm is getting enlarged. They getting elongated. And next, after that, intercept a cross wall forms, membrane invasion. The wall is getting invasinate like this. They grow like inwards, and a cross wall is formed here from the upwards and the downwards like this. They grow like this. And next, in fourth step, cross wall forms completely. The cross wall forms completely like this in the fourth step. After that, daughter cells in fifth step. We in this, if the this is the cell, nucleoid is present in this. After that, the nucleoid replicates itself, and then the cell wall is getting enlarged, cytoplasm is getting enlarged, and the inside nucleoid is getting separated from each other. After that, the cell wall is invasinate into the inward. After that, the cell wall is completely formed. After that, the daughter cell divide, and they develop into a single bacterial organism. This is the reproduction. And next, general characters of archaea, differentiating them from bacteria. Some of these share maximum amount of similarities, but they possess several distinctive characters, their cellular structure. Archaea have unique cell membranes composed of ether linked lipids, whereas bacteria have ester linked lipids in their cell membranes. Their cell walls lack peptidoglycan, which is a hallmark component of bacterial cell walls. Instead, some archaea have cell walls made up of pseudopeptidoglycan or various proteinaceous substances. In genetic makeup, archaea have genes and metabolic pathways that are more closely related to the eukaryotes than the bacteria in certain aspects. And next, their DNA replication, transcription, and translation mechanisms exhibit notable differences from those of bacteria. And next, the third one, metabolic diversity. These archaea can thrive in any extreme conditions, like the highest temperatures and highest salinities and acidic mediums and the alkaline conditions. Many archaea are chemotrophic, which means they derive energy from the various chemical reactions, including the methanogenesis, which means also called as the methane production. In this process, they undergo some process, and they produce the methane gas. And the chemolithotrophy, which means it is the oxidation of inorganic compounds. And the next one is a ribosomal RNA. The ribosomal RNA sequences of archaea differ significantly from those of the bacteria, contributing to their classification as a separate domain of life. And next, the histones. Archaea often have the histone proteins associated with their DNA, similar to the eukaryotes, which help in DNA compaction and the regulation. Next, the flagella. Archaeal flagella is structurally different from the bacterial flagella, both in the composition and the mechanism of propulsion. Propulsion means the way of the movement of its flagella. And next, the ecological role. Archaea play crucial roles in the various ecosystems, including the nutrient cycling, the methane production in the anaerobic environments, which means that there in anaerobic conditions there will be no oxygen, but in the absence of oxygen they produce the methane gas, and some undergo the symbiotic relations with the other organisms. In this we can observe, this is the picture of the archaea bacteria. This is the cell wall, cytoplasm, this is the nucleus, this is the dots we can observe in the cytoplasm are called as the ribosomes. And next, this one is the plasma membrane, this is the cell wall, and this is the capsule. The small hair-like structures is called as the pilus, and the tail region is called as the flagella, which helps in the movement, and in the movement called like propulsion. And next, the general characters of virus. These viruses are the unique infectious agents that straddle the line between living and the non-living entities. Here are the general characteristics. First one, nature. Viruses are obligate intracellular parasites. meaning they cannot replicate. They cannot replicate inside the host cell. If some viruses are outside the cell, they cannot replicate outside the cell. They can only replicate inside the host cell. They lack cellular structures and cannot carry out metabolic processes on their own. Next, the composition. Viruses consist of genetic material like DNA or RNA surrounded by a protein coat called as capsid. Some viruses have an additional lipid envelope derived from the host cell membrane. Next, the size. It differs from the ranging from 20 nanometers to the 300 nanometers in diameter. And next, the host specificity. Viruses exhibit a specificity for particular host organisms, tissues, or cell types. This specificity is determined by the interaction between the viral surface proteins and the specific receptors on the host cell surface. It differs between how the virus, how the proteins on the viral surface, and also the specific receptors which is present on the host cell surface. Next, diversity. It exhibits a wide range of structural forms, includes the helical structural, isohedral structure, complex structures, and the enveloped structures. This is the isohedral, and this is the enveloped structure. This is the helical structure. We can answer this is the helical structure, and this one is the more complex structure, so is called as the like TMV.